Good morning, students. Today we are moving to next section, that is polysaccharides. What are polysaccharides? They are polymerized products of many monosaccharide units. They are polymerized, combined products of many monosaccharide units. That's polysaccharides. Okay. Then polysaccharides are of two types: homopolysaccharides and heteropolysaccharides. Homo itself means that it is composed of single kind of monosaccharides. At the same time, heteropolysaccharides are those composed of two or different type of monosaccharides. In that, at least two or more than two saccharide units, sugar units will be different. Examples for homopolysaccharides are starch, glycogen and cellulose. And that of heteropolysaccharides, examples are hyaluronic acid and chondroitin. Starch is an example for homopolysaccharides that we are going to see now. Structure of starch. What is the significance of starch? It is a reserve carbohydrate in plants. In plants, starch is a reserve food. Okay. Then which are the sources of starch? Where all we can find the starch? It is present mostly in potatoes, tapioca and cereals. What about its composition? How they are forming? Using is a combination of which all monosaccharides? They are of amylose and amylopectin. 20% is of amylose and 80% is of amylopectin. What is amylose? Amylose itself is composed of many glucose units and they are linked through 1,4-glycosidic linkage. Is linked through 1,4. 1,4 means that glycosidic bond is forming between first carbon of one glucose unit and the fourth carbon of next glucose unit and forming a long chain. That is amylose. Amylose, its molecular weight is more than 400,000 Dalton or more. Amylopectin, it is the insoluble part. That means amylose is soluble. Amylopectin is insoluble. It is composed of again glucose units, but it is a branched structure, branch form. And that branching point, at that branching point, the bond that linking the glucose units is 1,6-glycosidic linkage instead of 1,4. Molecular weight of amylopectin is more than 1 million. Hydrolysis of starch. Iodine test is a very standard test to understand the presence of starch. If starch is present in a solution, if we add iodine, then that solution will become blue. That is a test for the presence of starch. So here in this test tube, to starch solution, iodine is added and it gives a blue color. Then if we heat that solution, what will happen? It will turn colorless, and then on cooling again this solution become blue color, because again that starch is forming, again that bonds are reconstituted and form the starch, and in between that or during that heating, it means that that bonds are break. Because of that breakage, that color lost their as the bonds again regain and reconstituted starch, again that blue color is reappearing here. So only when starch is present, the blue color will form. One more point we need to remember here, that starch is not reducing. Why it is not reducing? Because all glucose units are joined together one another. So there is no free, not much free aldose or ketose group. So that's why it's not reducing, and that is why if a starch is present, is giving blue color. As on heating, but that as that bonds are break, again that aldose and ketose groups are get freed, and it is becoming reducing. That's why it lost that color. Then hydrolysis by mild acids. On treating with mild acids, small fragments will produce small fragments out of the starch. Then if we continue that hydrolysis for much more time, for short time, first it will give amylodextrin, and that solution appears a violet color. That time it on reaction with iodine, because iodine is still there, and along with that we are hydrolyzing. So first product formed here is amylodextrin, and on forming amylodextrin, that solution turns violet color, and it is non-reducing. That means not much glucose units are free, not enough number of aldose or keto, free aldose or keto group are not available there. That's why it's not reducing. On continuing the hydrolysis for more time, next product forming is erythrodextrin. It gives red color with iodine. Then this erythrodextrin have a mild reduction ability to Benedict solution. If we pour Benedict solution to that starch solution mixed up with iodine, it will reduce. Reduce means it only mildly reduce. That means a few number of free aldose and ketos are available there. That it means. Then again, if we are continuing hydrolysis, then the next product form is acrodextrin. As acrodextrin is more hydrolyzed product, that means more bonds are broken, and more free aldehyde and more free keto group are present there. So it is reducing as all the most of the bonds are break, then it cannot react with the iodine. Then the last, even then, if we are continuing hydrolysis, then the next product can be formed is maltose. And as maltose is formed, we know that maltose formed of the combination by between two glucose unit. And here, as the chain is not that much long, it cannot give a blue color or any reaction with the iodine, and a small number of free aldehyde group is free, then it is a strong reducing agent also. Clear? Then action of amylose. Action of amylose on starch. Alpha amylose. How it act? Alpha amylose, amylase, salivary amylase and pancreatic amylase. These are two enzymes. One is produced from saliva, and another one is produced from pancreatic juice. They are all alpha amylase. Alpha amylase means the carbon that is involved in the bond is a configuration of alpha. That's why I'm alpha amylase. This alpha amylase act, how it act or where it act on starch. It acts on, this is alpha because they came I in place of El. Sorry. This alpha amylase act on 1,4 glycosidic bonds of starch in random position, not in any definite position. Randomly, it acts on the 1,4 glycosidic bonds of starch and convert to small units and break the starch to smaller units. smaller units repeatedly, and finally it become a mixture of alpha maltose. Alpha maltose means small fragments composed of two glucose units will be formed finally. Then beta amylase reaction. What about beta amylase? Beta amylase, it is present in plants, mainly in almond and germinating seeds. It usually breaks the starch to form beta maltose. Beta maltose, how the name came, I think you know. Then uh, action on amylose. How, what's the action? What's the nature of reaction here? It is split maltose units from one end because the starch is a long chain, and it starts splitting from one end, not in random position. Not in random position. Okay, that means from one end, it in one of its reaction, it remove one maltose unit, and in next step it remove another maltose unit from one end. Then again next step, again it removes another maltose group. In that way, it converts starch to small units. Then, what is the action of this beta amylase on amylopectin? Uh, that we are going to see here. Yeah, the action of beta amylase. It acts on amylopectin also. Amylopectin, we know that's a branched structure. But the action of beta amylase on amylopectin, it gets stopped at the points of 1,6 glycosidic bond. That bond is present at branching point. There, this enzyme cannot act. So, the result product is a large molecule, and it is called a limit dextrin or residual dextrin. Then glycogen. Glycogen, what is glycogen? It is a reserve food present in animals, not in plants. It is stored in liver and muscles. If we take the liver, its 5% weight is that of glycogen. And composition of glycogen, it is made up of again glucose units connected by alpha 1,4 glycosidic bonds and forms straight chain, and at branching point the bond will be, that linkage will be 1,6 glycosidic linkage. That is such a. Then in the glycogen, in its inner region, inner core present a protein called a glycogenin. Glycogenin is actually an enzyme that acts as a primer. Primer means something give a starting point, an initiation for the polymerization of glucose units, the linking of glucose units to one another continuously to form this glycogen. So, this glycogenin is an enzyme. It is found in the inner core. It acts as a primer, starting that initiational unit for the polymerization of glucose unit to give glycogen. It is more branched than amylopectin. Molecular weight is 5 million. This is the structure of glycogen. Here we can see the 1,4 glycosidic bond connecting the straight chain, and also 1,6 bond linkage at the branching point. Next is cellulose. Cellulose means it is present in plants. In cotton, 99% is of this cellulose, and in the case of wood, by weight, it forms 50%. The composition of cellulose, it is composed of glucose unit system by 1,4 beta linkage. This cellulose is forming glucose units with each other. These glucose units linked through beta 1,4 linkage. Beta 1,4 linkage means that anomeric carbon involved in bonding is that of beta. and a bond formed with between first carbon and fourth carbon of that of adjacent glucose units. It forms straight chain. There is no branching. Molecular weight is 2 to 5 million and is digested by only cellulose is an enzyme present only plants, is not present in animals or human digestive system, so they cannot digest cellulose. But in the case of herbivores and termites, they can digest cellulose because in their intestine certain bacteria are present, intestinal bacteria. Those bacteria help to digest the cellulose with the help of cellulose. They possess that enzyme. What is the significance? This cellulose is the starting base substance for producing fiber, celluloids, nitrocellulose, and plastic. Cellulose structure is given here. It's a long chain of glucose units, one for glycosidic beta, one for glycosidic linkage is here, this one. Next is inulin. Inulin means it's a long chain of D-fructose units. Fructose, we know it's a ketose sugar. D means dextro. It's a long chain of D-fructose units linked by 1,2 linkages. Here it's a structure. 1,2 linkage. 1,2. This is the second carbon. It's the first carbon between first and second carbon that linkage is forming. First and second. And occurrence, where all we can see this inulin is as a reserve food, reserve carbohydrate. Is present in various bulbs, tubers like chicory, dahlia, dandelion, etc. We can find this inulin. Significance. What's the significance? It can be, it is usually using in clinical, in medical field to understand the renal clearance value and glomerular filtration rate. Both these are connecting with both these are some methods used to understand the proper functioning of kidney, whether the kidney is functioning properly or not. Renal clearance value means it is a method used to understand the rate of removal of a toxicant from the body of the patient in unit time. That also indicates how the functioning of kidney. Glomerular filtration rate means there's a normal rate is there per minute. This much amount need to be formed. This much amount of glomerular filtrate need to form. If that amount is not forming, that it indicates that kidney is not working properly. Then dextran. Dextrans means is a branched homopolymer of glucose units, and that linkage, different type of linkages are present. 1,6 bond is there, 1,4 bond is there, and 1,3 linkages are there. Here this one is 1,6, this is 1,3 linkage, and this is 1,4 linkage. And it is produced by microorganisms. Its molecular weight is 1 million to 4 million. And it is produced by, is already mentioned. What is the significance of dextran? It is used as an intravenous infusion, as it will not go out of vascular compartment, for the treatment of hypovolemic shock to increase the plasma volume. What do you understand from this? I will explain. Intravenous infusion. If a patient lost so much of his blood, then the doctors will administrate this dextran intravenously, means directly into his or her vein. And because it will not comes out from the blood vessel, it can as it can retain there itself, it will increase the that plasma volume in within a very few time span. So in that way, patient can be protected or prevented out of this hypovolemic shock. Hypovolemic shock means that reduction in the plasma volume, loss of blood. That plasma volume is reduced below a certain limit, heart cannot pump enough blood to different organs in our body or different parts of the body, and this that may lead to the death of the patient. In such conditions, intravenously doctors used to administrate this dextran to increase the blood volume, plasma volume. That is the significance. Then chitin. Chitin means it is present. It is found in exoskeletons of crustaceans and insects. It is composed of N-acetyl glucosamine with beta-1,4 glycosidic linkage. It's composed of N-acetyl glucosamine with what? 1,4 glycosidic linkages. Okay. So homoglycan is over. Next we are moving to hetero. Hetero means composed of different types of glucose units. Then there can that is the phase I mentioned here. There are polysaccharides containing more than one type of sugar residues. Examples we are going to see. First one is agar. Agar's picture is given here. It occurs in seaweeds. And its composition is composed of galactose, glucose and other sugars. What's the property? At 100 degrees Celsius in water, it dissolves. But as it cools, again it will set and form this jelly-like substance. What is the application? For culturing bacterial colony, it acts as a supporting media because bacteria cannot digest this media. So that's one application, and another one it again uses a supporting media for immunodeficient and immunoelectrophoresis. What is immunodeficient? That I will show you. Simply it is used to understand the concentration of antigen and antibody formed in a given sample. Immunodeficient is a technique for obtaining a precipitate. Precipitate means how it forming. If the number of antigen and antibody formed formed reach an equilibrium, it will form a precipitate by binding each other. The antigen bind with the antibody at their equilibrium when their number is equal to one another. Antigen's number is equal to antibody. At that state, they perfectly form bonding each other. Then that time a precipitate will form. That is the immunodeficient. If that precipitate is formed, then we can understand that enough number of antigen and antibodies formed in that solution medium. So this is the supporting medium formed of this agar. In this two wells are there, means two depression, so two pits that carrying antigen and antibody, and it is keeping aside for some time, and these two start to migrate each other and form bond each other, and it will continues till reach an equilibrium, and after reaching equilibrium a precipitate is forming, and it indicates that enough concentration of antigen and antibody is formed there. And the application is that through immunodeficient we can understand the concentration of antigen and antibody and also the purity of that formed antibody. That is immuno. Then immunoelectrophoresis is the same process, but for making that movement we are using electric field. That's the difference. For these two processes, agar is using as the supporting medium. Then agarose. Agarose means it is a mixture of galactose and 3,6-anhydrogalactose. It's a combination of these two. And what's its application? It is also again used as a matrix, a supporting medium for electrophoresis. Then we are coming to mucopolysaccharides. Okay. Mucopolysaccharides or Glycosamine glycans, GAG. What are mucopolysaccharides? They are again heteropolysaccharides composed of it contains that type of heteropolysaccharides contains uronic acid and amino sugars, and these groups again acetylate also contain this heteropolysaccharides. In addition to uronic acid and amino sugars, they also contain acetylated amino groups, sulfates, and carboxyl groups. What's the property? Because of the presence of so many charged group like this, they can attract water molecules and will produce some viscous thick solution. Mucoproteins. Mucoproteins, if it combined with protein, then that mucopolysaccharides. Sorry. What are mucoproteins? Mucoproteins means mucopolysaccharides in combination with the protein. Then it is called mucoproteins. Examples of mucopolysaccharides, they are hyaluronic acid, heparin, chondroitin sulfate, keratin sulfate, and dermatan sulfate. We are going to see the hyaluronic acid. Where it is found? It is found in connective tissues, tendons, synovial fluid found in joints. Then vitreous humor. Vitreous humor, the fluid present in eyes. Aqueous humor and vitreous humor. Then function for lubrication and joint. How it is forming? What's the composition? It is repeated units of this combination: N-acetylglucosamine, beta-1,4-glucuronic acid, and beta-1,3-N acetylglucosamine. These units repeats again and again to form hyaluronic acid. Then heparin. Heparin is we know that it's an anticoagulant that prevents blood clotting. Application. If we are taking blood for any in vitro studies, lab studies that aim to prevent clotting, we are using heparin. And also in vivo means inside the body, to prevent some or to eliminate thromboembolic condition. Thromboembolic condition means formation of some clot inside the blood vessels. That condition also we administrate heparin to prevent or to recover from such situations. What it is doing, that heparin activates antithrombin. Antithrombin means it is acting against thrombin. Thrombin is the protein involved in blood clotting. That means it supports clotting. Antithrombin, it is against thrombin. So this heparin activates antithrombin. By that way, prevent unwanted clotting. Then where it is present in liver, lungs, spleen, monocytes. In all these things, heparins are found. And commercially, heparin is prepared from animal lung tissues. Composition. It's also composed of repeated units of sulfate glucosamine, alpha-1,4-L hydronic acid. And this, what is this hydronic acid? Hydronic acid is an oxidized form of idose. What is idose? Idose is an epimer. Epimer, I already told you what is epimer. Recollect it. Epimer with respect to fifth carbon. Okay. Then sulfated heparin also present in tissues. The next mucopolysaccharides that we are going to see is chondroitin sulfate. Where it is found is as a main substance, ground substance of connective tissue of cartilage, bone, tendons, cornea, and skin. Composition. Here the repeating units are that of glucuronic acid, beta-1,3-N, acetyl galactosamine sulfate, beta-1,4. This units repeats again and again to form chondroitin sulfate. Then keratin sulfate. Keratin sulfate, what's the specialty? These are only mucopolysaccharides without uronic acid. All the others have uronic acid. Composition, repeating units of galactose and N-acetyl glucosamine in beta linkage. This is mostly found in cornea and tendon. Then dermatan sulfate. Its composition is composed of repeated units of L-hydronic acid and acetyl galactosamine in beta 1,3 linkages. It's found in skin, blood vessels and heart valves. Next, coming to glycoproteins and mucoproteins. Before that, we need to understand what is proteoglycan. Proteoglycan means their carbohydrate chain attached to a polypeptide. Polypeptide commonly form proteins, right? So, if a carbohydrate is attached to a polypeptide chain, then it become proteoglycan. Then what is glycoprotein and mucoprotein? What's the difference? Somewhat they are similar. Proteoglycan itself. Then how that difference occurs? If that amount of carbohydrate, its amount is less than 10 percentage, then we call that proteoglycan as glycoprotein. And if that carbohydrate content is more than 10%, we call that proteoglycan as mucoprotein. So, somewhat they are synonyms, means same thing with different name. Where all we can find this glycoproteins and mucoproteins in almost tissues, all tissues and cell membrane. And as a glycocalyx, as a coat over the cell surface also we can see these substances. What's the function of these glycoproteins or mucoproteins? Their functions as enzymes, hormones, transport proteins and structural proteins. Then what is glycophorin? Glycophorin is a type of glycoprotein present in the membrane of RBC. It's a transmembrane protein. Transmembrane means it spans the membrane. What's the carbohydrate part? Composition of carbohydrate part of glycoproteins. Which type of carbohydrates we can see in a glycoprotein? Glucose, mannose, galactose, N-acetyl glucosamine, N-acetyl galactosamine, arabinose, xylose, L-fucose, N-acetyl uramic acid. All these things we can see as glucose component, glucose parts in glycoproteins. Okay, with this we finish this chapter and today's section. Thank you.